Yeah, I'd like to thank the organizer for the invitation to talk. And I'm going to go at the middle of the talk to tell you what the acronym actually means. But before we go, I'm not going to repeat what had been discussed early this morning. That's this issue. So I'm going to use graphic representation of what we are dealing with. So one of the three people will suffer from some form of a pain condition, and the actual issue that we're dealing with is this. And yep. So you're looking at this issue of the opiate prescription drug that, according to one of a recent study by the public health from Harvard University, that it actually kills twice as many people as cocaine and 5 times as many people as heroin. And we have heard many discussions this morning about the tolerance of opiates that really create this situation that we are dealing with. That is, in this country alone, it costs, the pain problem costs $6 billion a year, and about 14% of Medicare goes into treating the conditions of chronic pain. And as the population aging, and we see an increase of this issue. And as Dr. Mao alluded to early this morning, that pain is a condition that we must have. So we already know the nociceptive condition and the inflammation and the neuropathic condition that all create a painful stimulation to one of a harmful environment. And so there's really a dichotomy here that we understand the molecular and the cellular mechanism of pain in the past 2 or 3 decades. And there's a great advance in understanding the mechanisms. And we know the receptors and pain pathways, as many of the talk have showed us the recent advancements. But there's really a stagnance in the drug development. And if you look at it, very few drugs that actually surpass the existing ones. So why is that? And the major limitation that we have experienced in the past 2 decades is that we always try to find drugs that match the receptors in the body. And so we always try to find small molecules that will target existing receptors in the body. And so we focus on alternating the complex interactions of sensory input in our system. And because of that, and because most of the targets of pain is in the central nervous system, so we essentially mess around the interactions of central nervous system. And so the side effects of addiction and the psychoactive side effects is always associated with it. So I will not go through the theory that has proposed in 1965 of this gate mechanism of pain. But suffice to say that the idea of pain gating is that there's an inhibitory interneuron. If you excite the inhibitory interneurons, that actually inhibits the transmission cell to send a signal to the brain where the pain is perceived, right? So we try to develop a method so that we can, this is in the dorsal horn. And so we try to develop a method that we can move this outside the central nervous system, and namely in the peripheral nervous system, if it's possible. So as we know that the inhibitory receptors are predominantly mediated by the either GABA-A receptor or glycine receptor. And so we have worked for many years. My group has been focused on glycine receptor, but many other groups have been working on GABA-A receptor. So there's great advance in understand the structural aspect of it. And because of that, we were able to rethink the possibilities. So here's how we look into the alternative approach in drug discovery. And instead of seeking drug-like molecules to match the existing receptors, we ask whether we can provide the body with the potential target of our choosing. And so in that way, we can focus on the root of the problem of pain. And so we focus on the peripheral nervous system. And there's three possible mechanisms that we can focus on. One is obvious, is if we can hyperpolarize the afferents. And that's not very easy to do, especially under conditions where there's inflammation and there's also neuropathic change of the afferents. So if the nerve is injured, it's very difficult to actually hyperpolarize it. So that so if this one doesn't work, there's other ways of shunting the sodium channel. So if that is possible, and you can also alleviate the pain. And the third possible way of doing it is actually doing something that's naturally already occurred in the spinal in the actual dorsal horn. That's the excitatory neurotransmitter release through the primary afferent depolarization. So these mechanisms can be done, actually you can bring it outside the central nervous system by designing receptors. So we're thinking about designing receptor in such a way it will be installed in the dorsal root ganglia, or we can install it in the terminal of the afferents. And so that it will reduce the signal that's transmitted to this central nervous system. So the new concept is install non-immunogenic surveillance chloride channels either to the CN data fibers or dorsal root ganglion. And I'm going to show you some new development in my lab that actually probably also can be installed to other excitable cells. And that will reduce the signal initiation, and hopefully that will reduce the hypersensitivity to pain. And we hope that these designed receptors will remain silent under normal physiological conditions, so it will not interfere with the normal nociception. And, but they can either spontaneously respond to inflammation-induced changes, such as acidosis, or they can respond to small molecules that are otherwise have no analgesic action. So if you think about it, if you can turn some metabolites of common food into action that will have analgesic effects, that will be really very nice. And so that's where something is on the horizon. I use this background to suggest that possibly receptor engineering to lessen inflammatory invoke the pain and hypersensitivity. That's what RELIEPH stands for. It's probably one new way of addressing pain. And currently in my lab, we focus on inflammatory pain. But potentially the method can be used for neuropathic pain, but we have not tested that scenario yet. So designing principle is to use a channel that's highly selective for chloride. And we want it to be normally closed. So we design a channel that will conduct selectively for chloride. But they are normally closed, and they do not respond to neurotransmitters such as GABA and glycine. And they can be activated by either the pH, so that's passively, if there's inflammation, if there's acidosis, the channel will open on its own. Or you can provide small molecules that will activate the channel. So the class one is the pH-gated channel, where it will spontaneously open under the condition of inflammation. And we are in the design, we hope we are able to titrate the response curve based on how acidic the tissue is. And so that will, in other way to say it, is that the channel will adjust itself based on the degree of pain. And the class 2 drugs that we designed is turning non-analgesic molecules into a painkiller. And they are small molecules of our choosing. So I will show you one example where you can actually design the bonding site of your designer channels that will respond to propamine, which is actually a common metabolite of food. And so you can design your receptors in pair with the ligand such that it will be activated by small molecules. So in the design, we didn't go from ab initio. So we actually borrowed something that nature already developed. So this is a human glycine receptor, transmembrane domain. And the very interesting thing happened in the evolution, which we don't know why, that there's no glycine receptor in the peripheral nerve. So the inhibitory receptors, there's a GABA-A, which happens in the peripheral and the central nervous system. But for glycine receptor, it's restricted in the central nervous system. So we thought maybe we can borrow that condition so that we use the glycine receptor transmembrane domain from human. Then we tether it with some bacteria triggering molecules, receptors. So the different color, this is human, this is a bacteria, and so we modulated the interface of it. So the red is bacteria, the blue is human, and this is the initial design. Initial design is not very powerful, but after modulating it, so this is the structure that the transmembrane domain of glycine receptor was solved by my group using high resolution NMR. So this structure is actually, it's quite accurate. And so it's a very different from the conventional structure determination by crystallization, because crystallization, the structure is dictated by the crystallization condition, which is very different from the physiological condition. So, but by NMR, on the other hand, you are able to use determine the structure at the room temperature or at the physiological relevant temperature. So the transmembrane domain structure that we solved capture a lot of things that actually turn out to be very relevant to the function of the channel. And the actual cellular portion of it, we used two different types. One is the pH-gated pentameric ligand-gated ion channel called the ELIC, or the GLIC. And another one is ligand-gated ion channel in the bacteria. It's also pentameric ligand-gated ion channel called the ELIC. So we picked those structures, and that was also solved by several groups, including Dr. Payton's group, with ligand bonded to it, so that we were able to fine-tune the receptor bonding pocket of it. So this is just to electrophysiology data to show that the designed channel is indeed selective for chloride over sodium and other cations. So this is chloride selective channel. And this is class 1, which is passive. It does not require any ligand. It's only respond to pH. And as you can see that by changing only one amino acid in the TM2 domain, which is the pole nodding domain of the glycine receptor, we were able to change the pH dependence. For example, the initial design has this pH dependence around 7, so the EC50 is about 7.4. That means in the normal physiological condition, the channel is already opened. So that's not ideal. You don't want to have a channel that interferes with normal nociception. So that's not good design. And this is another design which opens at the pH 6.1. It's probably a little bit overkill. That means at the normal acidosis condition, under normal inflammatory condition where the the pH usually goes to 6.8. So this channel will open slightly. But looking at this one, which at the pH 6.8, it will open about 30 to 40%. So that's a way that you can titrate amount of response of your designed receptor to the inflammation. And the second class is to use small molecules that will activate the channel. So for example, in the design because we solved the crystal structure of the actual cellular portion of it, we were able to find a binding pocket for propamine. And we were also able to change the residues that are lining the binding pocket so that we move this dose response curve to the left. And as you can see that initially the propamine dose response is about 0.7 millimole, so that's 700 micromole, which is a little bit too high. And we were able to, by making these two mutations, to move the curve to the left, so it's 1.9 micromole of propamine that will open up the channel at 50%. So that's a very, very potent response to propamine. And so once we designed this, we said, let's test it in vivo. And the first model that we used is a model that people have developed and used for many, many years. It's the complete fluent adjuvant inflammation. So you inject the CFA to the paw, then you measure either the mechanical sensitivity by Fanfrey or the thermal sensitivity by the Hargreaves test. And this is to show that when you inject the expression vector to the paw, and this is just the plasma injection, you can actually detect the fluorescence signal, because in our design we have a portion of it that fluorescence is GFP, so that you can see in the DRG the engineered receptor is actually expressed. It's also showing up in the hind paw, in the nerve terminals. And we also package our designed receptor into this recombinant adeno-associated virus, and particularly this RELIEPH. When you inject it intrathecally, it expresses only, so you can see that this is the motor nerve fiber. It does not express our protein, but the nociceptors is expressed. So we don't know why. The RELIEPH apparently has selectivity preference for the nociceptor, for the sensory nerves, not the motor nerves, which is nice because you don't want to have your engineered receptor to numb the peripheral nerves. And this is just to show you that indeed our expressed proteins are colocalized with markers of nociceptors. So in the CFA model, and this is what happens. When you inject the CFA, so this is the Fanfrey test, and the animal responds to a force that's about 30 grams of force. And when there's an inflammation, they are going to respond to mechanical stimulation at a much lower level. And this green curve is the one that does not have any treatment. But when the animal express our engineered receptors, it did not bring it all the way back to the control level. So that means in the mechanical sensitivity, our engineered receptor does not alleviate the pain completely, but it does reduce the pain significantly. And it also recovers, the animal recovers much quicker than the ones that does not have our engineered receptors. But in terms of thermal response, it actually overshoots slightly. So this is the latency when you shoot a hot beam of light to the paw. And when they have inflammation, they pick up the paw very quickly. But this is on day one, second day. So when the inflammation is very severe, it still have hypersensitivity to thermal stimulation, but it recovers much quicker with the receptor. So these two tests are more or less invasive because you actually poke the animal. And so we ask whether we can actually measure the pain sensitivity to the thermal pain in a native environment, in an environment that's not intrusive. So we designed a plate, 2 plates. One is hot or cold. Another is room temperature. The animal cannot tell the difference of the plates. They are just next to each other with a very, very fine insulator between them. Then you look at the animal circling around in the cage on the plate where there's automatically recording of their location. And so when the plate is 30 degrees, 30 degrees, as you can see that the animal probably roughly spend 50% time on each plate. It's just running around. So this is the time spent on the non-30 degree plate. And if the plate is 8 degrees cold, animal really only spend 20% of time there, 80% time on the room temperature plate. But if the plate is hot, they really do not like the hot plate. They do not come out. But if you have our engineered receptor installed on the paw, this is even before the CFA inflammation, they venture out slightly more. It's 25 to 30% of time going to the hot and cold plate. But now this is after CFA. So one of the paw has inflammation. So that paw will dictate the behavior of the animals. And if you have installed your engineering receptors, but you do not have the ligand to trigger it open, the animal does not like cold when they have inflammation. They come out slightly on the hot plate. But look at when you put propamine to activate the engineered receptor, they actually indeed come out significantly more. That means there's an analgesic action coming, anti-inflammatory pain. And if you use the empty vectors, and the animal does not have any analgesic action. So now, how much time do I have? Probably, I have 3 more minutes. 3 or 4 more minutes. Yeah, so now I want to show you another example of inflammatory pain, and which is interstitial cystitis. As you know, that there's really no good treatment for interstitial cystitis. It's really a painful condition for many patients. And the current treatment is actually a Botox injection into the wall, the lining of the bladder. So we said that probably this method that we developed, because bladder is accessible from external, so you can instillate the expression vector into the bladder and have the bladder pick up the expression vectors. So in the animal study, we use this metabolic cage where you can measure the frequency of void and also the volume of each void based on the weight of urine that's being collected. And so then we can trace 24 hours in this metabolic cage of the animal behavior. And there's 2 models. One is control. Another is we use the cyclophosphamide, which will induce inflammation in the bladder. And so that's the red, is a CYP. When you inject that, there's a bladder inflammation. And as you can see that the number of void within 24 hours increased dramatically. And so each void, the void per volume is reduced. And another measurement that we can do is the cystometry, where you can inflate the bladder. And so there's inflammation in the bladder. So this is the curve. So when you slowly increase the pressure, and at a threshold, it contracts, and then it urinates, then it goes on like that. So there's a period between this. If you have your constant pressure inflate the bladder, and then there's a so-called intra-contraction interval. That's a measure of how sensitive or hypersensitive the bladder is. And as you can see that the normal situation, the intra-bladder contraction is about 400 seconds at the rate that we inflate the bladder. But when the bladder is inflamed, and this interval is reduced dramatically. So that's a measure of how sensitive the bladder is. And so in collaboration with John Beckel and Chet Gould, they have studied this problem for many, many years. So what they have found was that even the urothelial cells are excitable cells. And so even they are not nerve cells, they act like nerve cells. And they have this voltage-gated calcium channels. And as you can see that if you use voltage-gated, if you use the agonist of voltage-gated calcium channels, you're going to see increase of intracellular calcium, free intracellular calcium. And if you use the calcium channel blocker, it will reduce the intracellular calcium response. And the neurotransmitter, if you will, of the urothelium is actually ATP. So you can increase the ATP release. And by using the agonist of calcium channel, voltage-gated calcium channel, you can increase these contraction intervals. And so we actually look at the expression of our receptors. As you can see that the innervating L1, L2 in the rats, and also the L6 and S1 is overexpressed of the GFP signals. So this is the last slide that shows that our engineered receptor, which is in this yellowish bar, does not really change the underlying physiology of the animals when there's no inflammation. The voice per hour is pretty much the same as control. And the void per volume, the volume per void is also the same. But if you look at this trace, when you have a normal condition, the interval is like this. When you have inflammation, the interval is greatly reduced. But when you install our engineered receptor into the bladder, even before you put agonist to activate it, it's almost returned to normal contractility interval. And if you gave the agonist that activate our engineered channels, it actually overshoots slightly. So that shows that this method actually works in this interstitial cystitis model. So potential application in human, we are envisioning that, by installing selectively parallel nerve injection of the engineered receptor to the nerves that are affected, you probably can produce a situation where the patient will come to clinic once a year and receive an injection, then they are pain-free, or at least with reduced amount of pain for certain conditions. So with that, I think the RELIEPH method is long-lasting, non-psychoactive, and you can turn non-analgesic molecule into painkillers. And the conventional method, as we know, there's a side effects of dependence. And these are the people who did all the work. This is current group. This is a group a few years ago. Thank you very much for your attention.